Hello, everyone. Thank you for joining this session where we will be diving into the ICH Q1D Guideline, which addresses the application of bracketing and matrixing designs for stability testing of new drug substances and products. This guideline plays a crucial role in streamlining stability studies while maintaining compliance with regulatory expectations and ensuring patient safety. To set the context, ICH, or the International Conference on Harmonization, is a collaborative initiative involving regulatory authorities from Europe, Japan, and the United States. The ICH Q1D Guideline is part of a broader framework to harmonize technical requirements across these regions. Specifically, Q1D focuses on the proper use of bracketing and matrixing, two strategic approaches to reduce the scope of stability studies without compromising the quality or reliability of data. This guideline complements the broader ICH Q1A-R, which lays the foundation for stability testing as a whole. The key objective of this guideline is to recommend when and how reduced study designs can be implemented while still ensuring robust stability data. Essentially, it allows companies to minimize the number of tests by targeting only the most informative samples and time points. It applies to both new drug substances and finished pharmaceutical products. The guideline provides not only principles, but also practical examples to illustrate its application. Let's briefly compare full and reduced designs. In a full stability study design, every combination of product variables, such as strength, container size, and batch, is tested at every designated time point. While comprehensive, this approach can become resource-intensive, particularly for complex products. Reduced designs, including bracketing and matrixing, allow us to limit the number of tests by selecting representative samples or time points, provided that these reduced designs still yield sufficient information to accurately predict product shelf life and retest periods. Reduced designs are generally applicable to most conventional drug products, but caution is warranted for more complex drug delivery systems. These systems may involve intricate drug-device interactions that require a more comprehensive evaluation. For drug substances specifically, matrixing often offers limited value, and bracketing may not be appropriate due to the need to fully assess chemical stability across all specifications. Bracketing is a design strategy where only samples at the extremes of key design factors are tested. For example, the highest and lowest dosage strengths or the smallest and largest container sizes. The underlying assumption is that intermediate configurations will perform similarly to the extremes. However, it's important to justify why the selected extremes truly represent the worst-case scenarios for stability purposes. Bracketing can be applied across several factors. Dosage strengths that have similar formulations or manufacturing processes. Container sizes where one parameter varies, but the closure system remains constant. Closures, provided that their differences, such as vapor or oxygen permeability, are scientifically justified with supporting data. It is critical that significant formulation or packaging changes between variants are avoided, as these can compromise the validity of bracketing assumptions. Here's a practical example from the guideline. A product exists in three strengths: 50 milligrams, 75 milligrams, and 100 milligrams, packaged in 15 milliliters, 100 milliliters, and 500 milliliters containers. Instead of testing all combinations, the study tests only the lowest and highest container sizes across all strengths. By focusing on the most extreme scenarios, we can substantially reduce testing without sacrificing meaningful data. Matrixing takes a different approach. Instead of focusing on extremes, it distributes testing across different combinations at different time points. Not all combinations are tested at every interval, but all are tested at key stages. This allows for a significant reduction in testing burden while still capturing the necessary information about the stability profile over time. In a well-designed matrixing study, all factor combinations must be tested at the beginning after 12 months and at the final time point. At least 3 time points, including baseline, are required during the first year for each tested combination. Separate matrix designs must be used for each storage condition, such as long-term, accelerated, and intermediate storage. These precautions ensure that even with reduced testing, sufficient data are gathered to support accurate shelf life estimation. The guideline provides several examples of matrixing designs. 1/2 reduction, where about 50% of the full testing plan is executed. 1/3 reduction, where roughly 1/3 of the tests are performed. However, after factoring in required full testing at specific time points, the actual reduction is typically less than these theoretical percentages. In some cases, designs can also combine both time point and factor reductions. While matrixing offers flexibility, its appropriateness depends heavily on the nature of the product. For products with predictable and consistent stability profiles, matrixing is often acceptable. If stability data show moderate variability, statistical justification becomes critical. For highly variable products, full designs may be necessary to ensure reliable shelf life estimation. Overly aggressive reduction can risk generating insufficient data, potentially leading to regulatory rejection. Importantly, stability data generated from reduced designs are analyzed in the same way as full study data. The statistical models used must still provide reliable estimates of product stability. If changes occur during the study, such as switching from reduced to full testing, statistical adjustments are required to maintain data integrity. To summarize, bracketing and matrixing designs are valuable tools for efficient stability testing. They reduce testing burden without sacrificing data quality, provided that they are scientifically justified. Ultimately, these designs enable organizations to manage resources better while fully complying with regulatory expectations and ensuring patient safety. The guidance we reviewed today comes primarily from the ICH Q1D Guideline, supported by the broader ICH Q1A framework, which remains the foundation for stability testing requirements. That concludes the formal part of today's presentation. Thank you for your attention. I'd now be happy to take any questions in comment box you may have.